Okay. Next, we are going to the functions of the vitamin K. The biological role of vitamin K is to act as a cofactor or coenzyme for a specific carboxylation reaction that transforms selective glutamate residues to a gamma carboxyglutamate residues. The reaction is catalyzed by a macrosomal enzyme, a gamma glutamyl, or vitamin K dependent carboxylase. The four vitamin K dependent procoagulants, factor 2 or prothrombin, and factors 2, 9, and 10 are synthesized in the liver and then secreted into the circulation as zymogen. Zymogens is inactive forms of them. Their biological activity depends on their normal complement of GLA residues. The GLA is the gamma carboxyglutamate residues. GLA residues is the a certain protein that contain the residues, namely gamma carboxyglutamate, which are efficient chelators of calcium ions. Two other vitamin K dependent proteins, such as protein C and S, play a regulatory role in the inhibition of coagulation. The function of protein C is to degrade phospholipid bound activated factors 5 and 8 in the presence of calcium. Protein S acts as a synergistic cofactor to protein C by enhancing the binding of activated protein C to negatively charged phospholipids. Vitamin K dependent proteins can be found in bone, cartilage, kidney, lungs, and other tissues. And the example, osteocalcin and matrix glycoprotein. Okay, this is the diagrams of the epocyte cycle of the vitamin K. The cycle metabolism of vitamin K in relation to the conversion of glutamate. This is the glutamate from the glutamate to the GLA residues. GLA residues, the certain protein which contain the gamma carboxyglutamyl residues in its tail for the coagulation of protein prothrombin. The active form of vitamin K is the vitamin K quinol or vitamin K hydroquinone. Needed for carboxylation is the reduced form the vitamin K quinol. This is the vitamin K quinol. The carboxylation reaction is driven by a vitamin K dependent carboxylase activity which simultaneously converts vitamin K quinol to vitamin K2 triacoxide. Vitamin K2 triacoxide is reduced back to the quinol and then to the quinol by the vitamin K epoxide reductase. The reductase activity denoted reaction 2 is the thiol dependent and is inhibited by co-primary anticoagulants such as warfarin. Dietary vitamin K, vitamin K quinone we can get from the dietary sources may enter the cycle via an NADPH dependent or NADH, nicotinamic acid dehydrogenase, dependent vitamin K reductase activity, which is not inhibited by a warfarin. Okay, uh, what's the factors affecting the vitamin K requirement? First one, bioavailability of vitamin K from foods. It has been estimated that the efficient of absorption of piloquinone from boiled spinach eaten with butter is not greater than 10% compared with an estimated 80% when piloquinone is given in its free form. or from supplement. This poor absorption of piloquinone from green leafy vegetable may be explained by its location in chloroplast and tight association with the thylakoid membrane where the naphthoquinone plays a role in the photosynthesis. In comparison, the bioavailability of menaquinone from butter artificially enriched with this vitamin was more than a twofold higher than that of piloquinone from spinach. Fats and oils that are contained in mixed diets were found to make an important contribution to the piloquinone content of the US diet, and in a UK study contributed 30% of the total dietary intake. And the second one, infection, for the because the malabsorption syndromes can inhibit the absorption of vitamin K. And the third one, the drug nutrients interaction like the warfarin, because the warfarin can inhibit hepatic vitamin K apophyte reductase activity. And how about the vitamin K metabolism? The first one, the absorption and transport. Dietary vitamin K, mainly piloquinone, is absorbed chemically unchanged from the proximal intestine after solubilization into mixed micelle composed of bile salts and the products of pancreatic lipolysis. Within the intestinal mucosa, the vitamin is incorporated into chylomicrons, is secreted into the lymph, and enters the blood via the lacteals, the lymphatic capillaries. Once in circulation, piloquinone is rapidly cleared at a rate consistent with its continuing association with chylomicrons and the chylomicron remnants, which are produced by lipoprotein lipase hydrolysis at the surface of capillary endothelial cells. After an overnight fast, more than half of the circulating piloquinone is still associated with triglyceride-rich lipoprotein, with the remainder being equally distributed between low-density and high-density lipoproteins, although piloquinone is the major circulating form of vitamin K, like the menaquinone 7 is also present in plasma at lower concentration and with lipoprotein distribution similar to piloquinone. And this is the illustration for the absorption and transport. We can see this. Dietary vitamin K will absorb because of the mixed micelle. Vitamin K, like the K1 and menaquinone 7, along with mixed micelle, absorb to the brush border of the enterocyte and the intestinal space. The vitamin K along with a triglyceride, a phospholipid, a polypoprotein, a lipoprotein, and the other lipids will be packaged to the chylomicrons and the specific for the chylomicron has the you can see that has the apolipoprotein B48 and the apolipoprotein A in on the its surface. Chylomicron has the apolipoprotein B48 and apolipoprotein A on the surface. So this complex will move to the lacteal. Lacteal, you can assume it the mouse pit or the small road, yes, the small road, the complex move to the small road. The small road is the lacteal and going and goes to the mesenteric lymphatics. You can assume the mesenteric, you can imagine the mesenteric lymphatic as the highway. uh, okay, this complex you can imagine as the car. The car the car is going to go to the small road, the lacteal, and then to the highway. Highway is the mesenteric lymphatics. For the toll road. Toll road is the blood circulation via the toll gate. Toll gate is the thoracic duct. You can imagine thoracic duct as the toll road. Okay, I will repeat it. The car is going to go to small road. Small road is the radial, and then to the highway. Highway as the mesenteric lymphatics, because the car went to the toll road via the get tolls. Okay, get toll is thoracic duct, and next going to the circulation of blood, like the toll road. Okay, the car. Okay, the car now the car is at the blood circulation. Is the blood circulation for moving to the liver. Okay, and the car or the complex will enter to the bridge, the large bridge is the bad capillaries. You can see that there's the capillaries, and the capillaries is there is and the inside of the capillaries, they are the LPLs, lipoprotein lipase. But before enter the large bridge or the bed capillaries, the complex or the cars need a apolipoprotein from the HDL, like the apolipoprotein C and E. So it can enter to the bridge or capillaries, but in the capillaries, lipoprotein lipase degrade many triglycerides cargo, and degrades lipoprotein C and A. So that's why when the chylomicron exits from the large bridge, the chylomicron to be the chylomicron remnant, because it has only a lipoprotein E and B48. Okay, next the chylomicron remnants will continue its activity to the liver via the blood circulation. Okay, this is the describe of this diagram. Intestinal absorption of dietary phyloquinone K1 and MK2 in the intestinal lumen. K1 and MK2 are incorporated into mixed media comprising bile cells, the products of pancreatic lipolysis, and other dietary lipids. Mixed media are taken up by intestinal enterocytes of the small intestine and are incorporated into nascent chylomicron, which have apolipoprotein A and B48 on their surface. Chylomicron are secreted from within the intestinal villi into the lymphatic capillaries or reticules, which join larger lymphatic vessels and empty into the blood circulation via the thoracic duct. In the bloodstream, a chylomicron acquire a POC and a POE from HCl. Chylomicron enter the capillary beds of the peripheral tissue, where they lose much of their triglyceride cargo through the action of lipoprotein lipase, at the same time losing ApoA and ApoC. Okay, you can imagine lipoprotein lipase like a thief, like the thief, like the thieves that take the cargo from the passenger. The cargo like this, the triglyceride, apolipoprotein A and C. And next, chylomicron remnant that re-enter the circulation are smaller and have a central lipid core with surface apolipoprotein B48 and ApoE. Okay, next, the mechanism of cellular uptake of vitamin K. Uptake of the vitamin K by the liver, the majority of vitamin K is delivered to the liver within the CR generated during the postprandial phase of intestinal absorption, and this is the continue from this previous figure, our previous illustration, the chylomicron remnants with the apolipoprotein E and B40X enter to the liver, uh, to the liver, and then the uptake process is complex and involves different proteins on the surface of lipoprotein cell surface, low affinity binding sites of heparin sulfate proteoglycan, and high affinity lipoprotein receptor that mediate internalization of the lipoprotein particles. One important receptor component is ApoE, apolipoprotein E. The primary relevance of the ApoE to the transport and slow internalization vitamin K is that it acts as a ligand that facilitates the high affinity binding of lipoprotein to the LDRR and other members of the LDRR family, which include the RRP and the VLDLR, the receptor of the VLDL. Okay, we can see this. The car or the complex, the chylomicron remnant can enter to the liver because of the apolipoprotein E and B48 is recognized by the RRP and LDRA. Okay, the RRP and LDRI will say to the chylomicron remnant, okay, access is accepted because the receptor recognize this apolipoprotein E or B48. So the vitamin K can enter to the liver to metabolism and secrete to the bone or to the other tissue or the organelle cells and etc. Okay. Uh, will continue the vitamin K, uh, vitamin K. Next, the vitamin K. After the vitamin K is metabolized by the liver. Next, the liver will be secreted the vitamin K to the other organelles, to the other tissue or bone, along with apolipoprotein B100. Apolipoprotein B100 is the specific for the VLDL. So the vitamin K with its transport going to are going to the bone via the blood circulation, and in the journey, is the circulation. It needs acquire the apolipoprotein C for the requirement to enter to the capillary in the peripheral. So this inside the capillary has the thief. The thief is namely the LPR, lipoprotein lipase, and lipoprotein lipase degrade some triglycerides and the apolipoprotein C. After that, after this action, and next the chylomicron to be the not chylomicron, but by VLDL to transform to be the IDL because some triglyceride had degraded by the lipoprotein lipase, so VLDL transformed to the IDL. For the IDL, the vitamin K has its transportation, namely apolipoprotein B100 and apolipoprotein E, and the apolipoprotein C has the taken up by the LPL. Okay, LDL to be will transform to be the LDL because LPL action. So the vitamin K. So now the vitamin K just has the apolipoprotein B100 as the key, or the ID card to the requirement for the entering to the bond for the bond matrix. Okay, let's see the vitamin K with its transportation, like the apolipoprotein B100 will enter to bond matrix, but before it enter, LDL receptor must be recognized its transportation. Okay, when the LDL receptor see the vitamin K and apolipoprotein B100, the LDLR recognize the B100. So LDLR will say to them, okay, access is accepted and you can enter to the bond matrix and welcome. Something like that, yes? You can imagine like the James Bond wants to enter to the room with the many password, and the gate will be opened if the James Bond can give or can provide, can say the right password or right key. So the gate, like the receptor, will say, okay, access is accepted. Next, so the vitamin K will serve its functions to the bond matrix, like the mineralization or the resorption of bond. Okay, it's likely that a major fraction of dietary K1 and MK2 is delivered to the liver and bound within the chylomicron remnants, which have ApoB48 and ApoE on their surface. CR, or chylomicron remnants, can interact with the cell surface lipoprotein receptor, example LDLR and LRP, and are taken up by target cells by receptor-mediated endocytosis. There's evidence that K1 and especially MK7 are incorporated into LDL through the hepatic export of VLDL bearing surface ApoB 100C and E, followed by their subsequent dilapidation in capillaries to VLDL remnants, IGL, and further catabolism to the LDL, and the LDL has ApoB 100 on its surface. It is known that human osteoblasts express the LDLR and RRP1, and that RRP1 plays a predominant role in the uptake of CR by osteoblasts, including the ability to utilize K1-rich CR for a gamma carboxylation of osteocalcium. And the vitamin K is excreted by a bile, and the vitamin K is essentially metabolized in the liver and excreted in the urine and bile. Okay, for the as much as 20% of pelocin was recovered in the urine, and as much 40 to 50% was excreted in the feces via the bile. Okay, what will happen if the person excess of vitamin K and deficiency of vitamin K? For the hypervitaminosis, no adverse effects associated with vitamin K consumption because vitamin K is rapidly metabolized and excreted via the urine or the fecal because of bile action. So no adverse effects of the vitamin K. And how about if the person deficiency or lack of the vitamin K? Okay, the person will occur the bleeding and hemorrhage and osteoporosis, and the people can feel the osteoporosis because reduced bone mineralization and caused or the lead to the osteoporosis.